Hi, I'm Ben,
I'm Collin.
I'm Sara.
and this is our presentation on a process developed by Firmenich. Firmenich is a private Swiss company in the flavor and fragrance market. They produce fragrances, perfumes, flavorings, and home care products. Firmenich also owns approximately 13.5% of the global market share in this industry. Two chemicals that are commonly used for fragrances are patchoulol and 7-epi-alpha-celanine. They are both naturally derived from valerian oil, which comes from the Valeriana jaffamonsi plant. The price and availability of these chemicals are affected by the season, the oil yield that year, the origin of the plant, the abundance, and local conditions. Collectively, these factors make it almost impossible to acquire patchoulol and 7-epi-alpha-celanine sometimes. As you can see, synthetic processes are quite valuable. Companies already have a method for individually developing each of these chemicals via a synthesized enzyme or RNA. But Firmenich sought to have a process that produces both patchoulol and 7-epi-alpha-celanine with little waste and more energy and resource efficiency.
The general idea of this process is to isolate DNA from a specific plant and place it into E. coli bacterium. The gene itself will create pathways within the bacteria that allow it to produce enzymes which catalyze the transformation of farnesyl pyrophosphate, also known as FPP, into the more useful chemicals patchoulol and 7-epi-alpha-celanine. Both of these products play important roles as products for Firmenich. Specifically, the process starts with scientists obtaining the RNA of the Valeriana jatamansi, also known as the Indian valerian. They do this by crushing the plant and running it through the concert plant RNA reagent, which separates the RNA from the rest of the cells. After purifying the RNA, it is run along an agarose gel via electrophoresis, which separates the individual genes by length of the strand. The useful cDNA can then be excised from the gel, at which point it is further cloned to obtain significant amounts of the relevant gene, known as Val-R20. At this point, the Val-R20 is added to a ligator along with plasmids. The gene is incorporated into the plasmid and then added to an inoculator. E. coli are also added along with a medium in which they can grow. In this environment, the plasmids get incorporated into the E. coli, so the gene is within the DNA strands of all the E. coli within the sample. Afterwards, the products are run through a centrifuge for 30 minutes at 18,000 G, which causes the E. coli to die and the enzymes to separate from the host bacteria. The enzymes are then added to glass tubes along with MgCl2 and farnesyl pyrophosphate. They help catalyze the transformation of FPP into 11 products, including patchoulol and 7-epi-alpha-celanine. By lowering the activation energy for the chemical transformation of FPP into patchoulol and 7-epi-alpha-celanine, the entire process can be easily controlled and the rate of reaction rises dramatically. Without the enzyme, the FPP could degrade into any number of worthless byproducts. The product, now referred to as Clearwood, recently won SEPOA's 2015 award for innovation.
The unit operation that captures the unique technology for this company is the incubator that is used after the inoculation process. Inoculation is the introduction of cells or organisms into a culture medium. In this process, the inoculation of 5 milliliters of LB medium is done using single colonies of E. coli cells. LB is lysogeny broth. LB is very rich in nutrients and is commonly used to grow bacteria. Once an OD of 0.3 is reached, the cultures are used again to inoculate. However, it is a total of 25 milliliters of LB inoculated. OD is optical density, a method used to estimate concentration of bacteria in liquid. The bacteria is then placed in the incubator at 37 degrees Celsius. In this incubator, the OD reaches 0.5. Next, the bacteria is placed in a 20 degrees Celsius incubator and left overnight. This is a 0.2 increase in optical density, which at such a scale correlates to a linear increase in bacteria concentration. This allows for a greater product yield and reduces the amount of waste. The most important part of this operation is having discovered the perfect temperature to grow bacteria at an optimal rate.
Thank you for listening.